Hello students, today we are going to start the last type of connective tissue, that is fluid connective tissue. Earlier we have studied that there are three types of connective tissue proper or loose connective tissue, dense connective tissue and specialized connective tissue. Under the specialized connective tissue, we have studied the skeletal connective tissue. Under the skeletal connective tissue, we have studied the bone and the cartilage, right? After that, in the same, that is specialized connective tissue, which have another part that is known as the fluid connective tissue. The second part will be the fluid connective tissue. Under the fluid connective tissue, there are two types of fluid connective tissue. One is blood and another is lymph. Okay, so there are two types of fluid connective tissue. One is blood and the second one, that is lymph, right? Now after that, whenever it comes to the fluid connective tissue, we should know that both blood and lymph, they are flowing in separate vessels. If it comes to the blood, that will flow inside the blood vessels, and the lymph, that will flow in the lymphatic vessels. Both of them, that will be circulating throughout the body, okay? And in the body, they will basically circulate the nutrients, gases, carbon dioxide, nitrogenous waste product, hormones, etc. Right? So first of all, we will see the blood, and after that we will see the lymph. So first of all, let's just start with the blood, right? So what is blood? That is the fluid connective tissue, and the blood is actually connecting throughout the body, and it is the softest tissue of our body also. One healthy individual can have around 6 liters of blood, and that blood, it will be not consistent in our blood vessel. From the blood vessel, some amount of blood can squeeze out and it will form the tissue fluid that later on will convert into lymph. And once again, the lymph will connect to the blood vascular system. So we will come to that part whenever we will proceed to lymph. So first of all, let's just see some introductory point about the blood. So this blood is the soft tissue that runs inside the blood vessels, right? Remember, the blood will flow in the blood vessel. There are three types of vessels present. One is the artery, second one is the vein, and third one is capillary. In the arteries, normally the oxygenated blood flow, except the pulmonary artery, through which the deoxygenated blood flow. In the veins, normally the deoxygenated blood flow, and there is one exception, that is the pulmonary vein, and from the pulmonary vein, one oxygenated blood that will flow. And the last type of blood vessel is the capillary, which is the finest, thinnest blood vessel, which will actually circulate the mix, that means both oxygenated and deoxygenated blood. Now, first of all, we should know what is the color of the blood. The color of the blood is red, and here the most abundant cell is present because of what the color of blood is red is RBC. On the surface of RBC, there is a pigment present, and that pigment is hemoglobin. And because of the hemoglobin, the color of the blood is red. Okay, so the next important point we need to know that this blood will have a specific pH. What do you think? The blood will have acidic nature or basic nature, right? So it's actually a basic. It is slightly basic in nature. And what is the pH? Remember, the pH is around 7.4. Remember, it is basic in nature and its pH is 7.4. This blood that will transport the various components in our body, what are they? That will be gases, carbon dioxide, oxygen, then the nutrients, then waste product, hormones, etc. Right? Minerals also, electrolytes also, that will be also circulating. Now come the components of blood. Okay, so first of all we'll see the components of blood, right? So basically the blood is made up of two parts. What are they? Okay, so let's just see. If you classify the blood, then this blood will be of two types. One is the formed component, right? One is formed component and second one is liquid component, right? The liquid component will be known as, this liquid component will be known as the plasma. And what is that form component will be made up of? That form component will be of three types. Basically, they are the cells. How many cells are present in the blood? There are three types of cells. What are they? First one, this is RBC. Second one, this is WBC. And the third one, that is platelet. Right? RBC means it is erythrocyte, WBC means it is leukocyte, and platelets, that is also known as thrombocyte, right? So these are the three formed components are present. And if it comes to the liquid component, so that liquid component will be known as the plasma, right? So that will be known as the plasma. So this plasma will be that will be divided into two parts. What are they? One is the protein part, okay, and another component that will be known as the serum. So how many types of proteins are present? There are three types of protein. Remember, what are they? One is fibrinogen, second one that is known as globulin, and the third one that is known as albumin. Okay, so there are three types of proteins present. If it comes to the fibrinogen, what is the function of fibrinogen? Fibrinogen basically helps in blood clotting. So whenever the blood comes out of the blood vessel, suppose due to any cut, then after some time what will happen to the blood? That will get coagulate. Why this will happen? Because of fibrinogen. Second one is globulin. Nowadays also we are talking about immunity, right? So what actually immunity will be provided? That will be provided by some antibodies, isn't that? So these antibodies will be immunoglobulins, right? And that will be the defense protein of our body that will fight against the foreign particle or pathogen. That will be globulin. And the third one is known as the albumin. Now for this albumin, see, our blood will have a consistency, right? That means how much fluid it is, or what we can say that what is the viscosity of the blood? Is it more thicker or it is more dilute? Okay, so what is that? That fluidity will be maintained, that will be albumin. So this albumin will maintain the osmolarity of the blood. How much liquid water should be present in the blood, that will be decided by albumin protein. Okay, so these are the three types of components present in the plasma, and if you remove that protein part from the plasma, then the remaining part will be known as the serum. Okay, so that is the component of blood plasma. Now if you come to the form component RBC, WBC and platelets, then you can see that the WBCs can be further divided. The WBCs can be further divided into two types. One is known as granulocyte. Okay, one is known as granulocyte, and second one, this is known as a granulocyte, right? If it comes to the granulocyte, then there are three types of granulocytes present. What are they? One is eosinophil, next one is basophil, and the third one that is known as neutrophil. Okay, and if it is agranulocyte, the another type of WBC is agranulocyte, and there are two types of agranulocyte present. One is monocyte, okay, and the second one that is known as lymphocyte. Okay. Okay, so that will be very much important. Now I will give you a simple question so that you can understand that from this part how the question can be asked. Okay, so the question will be, suppose this is the question, plasma is equal to formed component minus serum. Okay, suppose next one, that will be blood minus proteins. Third option is blood minus formed component, and the fourth one, formed component plus protein. I will write here. Okay, and the fourth one, it will be blood minus RBC, WBC, platelet plus protein. Okay, so what will be the answer? So there are lots of questions that can be asked depending, and that format will be like this. That plasma will be equal to this minus this, serum will be equal to this minus this, or this plus this. This way question can be asked. Okay, so for that we will see a simple experiment by which you can easily answer, you can easily able to answer what will be the correct option from the above. Okay, so first of all, let's just see that experiment. Okay, experiment to separate the components of the blood. Okay, so first of all, in a test tube, suppose this is the test tube. In a test tube, we will take a amount of blood from a suppose person. We will take the blood in a test tube. So first of all, what will be the color of the blood? It will be dark brown, it will be dark in color. Okay, then first of all, what you have to do, first of all, add anticoagulant, because it is going to get coagulated outside the blood vessel. So that's why first of all we will put one anticoagulant into that that one. Then after that we will centrifuge it. Okay, so whenever we will centrifuge, okay, this test tube in 2500 rpm for 5 minutes. Okay, now what is centrifugation? Centrifugation means rotation. Okay, there will be a machine. In that machine, what you have to do, put that test tube. Okay, and you will have two buttons. One button will be for rotation, how many times this test tube is going to rotate inside that centrifugation machine. That will be known as RPM. What will be the full form of RPM? That is rotation per minute. So in 2500 times it will rotate in 1 minute for how many time? That will be for 5 minutes. So in 1 minute that test tube is going to rotate for 2500 times. So this way it will continue up to 5 minutes, and after 5 minutes that machine will be switched off, and you will see certain changes in the test tube. Okay, so what you have to do, that first of all centrifuge that test tube, okay, inside that centrifugation machine. Then after that you will get one difference that will be very much clear. What will be that towards the bottom you are going to see some crystallized structure. Okay, some crystallized structure that will be deposited on the bottom. Okay, suppose these are the crystallized structure that is deposited in the bottom. Then after that there will be a layer which will be transparent. It will be white, opaque or transparent, and above there will be a liquid layer. Okay, and that will be slightly, it is yellowish or reddish orange in color. Okay, so this way you will see certain layers. Understand? So what that layers actually will have. Okay, so what we will see on the bottom you will see that the two components will be present. One is the RBC and second one will be the platelet. Okay, next one, just above that, what we will see a transparent layer. Okay, so what is that transparent layer will have? It will be WBC. Okay, and above there will be a yellowish layer you will find. Now what is that? That will be known as the plasma. Understand? So this way we will have the three layers. Towards the bottom you are going to see RBC and the platelet. Above we will see WBC, and above there will be a liquid yellowish in color that will be known as the plasma. Right? Now just see, after that this test tube have to be, once again we will do one thing, that in another test tube we will take out the plasma. Okay, we will separate out the plasma. Okay, leaving that RBC, WBC and platelets, we'll just pipette out. With the help of a pipette we will take the plasma in a different test tube. So suppose we have separated the plasma here. Okay, so whenever we have separated the plasma in a different test tube, then once again we will centrifuge it. Okay, we'll once again centrifuge in 2500 RPM for 5 minutes. Okay, so whenever we centrifuge that test tube having the plasma only for 5 minutes, then what we are going to find. Okay, so just see, whenever you open that centrifugation machine, okay, it will rotate 2500 RPM for 5 minutes. Then after you remove the test tube outside, then you will see two different layers. Actually, whenever we centrifuge now, that due to the gravity, actually some the components which is heavier, that will get deposited sooner. Okay, so first of all what will happen, towards the bottom once again we will see some type of structures. Okay, that will be also crystalline, small globe like structural granule like structure that will be deposited, and above there will be a transparent layer. Okay, suppose this will be a transparent whitish layer that will be present. So in that plasma, whenever we centrifuge, then what will be seen once again. Okay, so just see, towards the bottom you will see this will contain the proteins. Remember I told that this plasma will have three types of proteins. What are they? Fibrinogen, globulin and albumin. So that fibrinogen, albumin and globulin that actually get deposited on the bottom whenever we centrifuge the plasma, right? So that are the three types of protein. What is the function of fibrinogen? That will help in clotting of blood. Second one is albumin, which will maintain the blood osmolarity. And the third one is globulin, which will actually the immunoglobulin, which will be a defense protein, which will kill the foreign particle. Okay, so that three protein component that get deposited on the test tube, and above what will be that layer which is now it will be not exactly transparent, but it is white or slightly it is opaque in nature. Okay, so what is this? This will be the serum. Okay, so that serum will be nothing but it's around 90% water. Along with that, it will have some amount of minerals, electrolytes, suppose potassium, sulfur, then calcium, sodium. That component, that ions will be present in also the blood. So it will be the composition of the serum. Then along with that, the dissolved gases, then the nutrients, the food particle, whatever we have taken, that is transported by blood. So in the serum, we will have this nutrient which is now in the biomolecule state, suppose glucose, fructose, then amino acid, then fatty acid, glycerol, that will be present. Okay, then along with that, some oxygen, carbon dioxide, that gases will also be present, and other components like hormones, that will be also there in that serum. Okay, so that will be the complete separation of the particles, components in the blood. So what we can say that the blood will be equal to, the formed component plus plasma. And what will be the plasma? If you remove the form component, okay, that is RBC, WBC and platelet from the blood, then you will get the plasma. Okay, so that will be the plasma after removing the RBC, WBC and platelets. But after that, what we can see, once second, when we can get the serum, that from the plasma, if you remove that protein particle, then you will get the serum, right? So what will be the blood? Blood will be equal to RBC, WBC, platelet, that will be the formed component, and plasma. What will be the plasma? Plasma will be equal to blood minus formed component. That means, isn't that if you remove that RBC, WBC and platelet, then you will get the plasma, right? So this will be the equation of the plasma. And the next one, what will be the serum? Serum will be equal to plasma minus proteins. Okay, so what are those proteins? Fibrinogen, albumin and globulin. If you remove the protein factors from the plasma, then you will get the serum. So that is about the experiment by which we can able to separate out the different components of the blood. Blood will be made up of the formed component, RBC, WBC, platelet and the plasma. Plasma is made up of the components, protein components, that are fibrinogen, albumin and globulin, and the serum. Serum will have water, minerals, ions, electrolytes, gases, nutrients, hormones, etc. Okay, so this is just the introduction of the fluid connective tissue blood. In the next video, we will see the components of the blood. Okay, so hope you have understood. That is about the introduction of the blood. Thank you.